online and recording to this talk on glue genes. Jonathan is going to talk about glue genes, so I will just talk briefly about Jonathan. So Jonathan works for glue genes and has been doing that since 2020, 2021. But 20 years ago, not today necessarily, but in this month, he graduated with a bachelor's in astronomy from Caltech, after which he moved over here to Harvard, where he did his PhD in astronomy also, then did a postdoc and was a lecturer at Boston University, after which he went to Yale for a postdoc. These are all sort of short postdocs. These are physics postdocs. They're like 1, 2, 3 years. They're not 5-year biology postdocs, just for context. And then he joined Hollywood, but as far as I can tell, remotely. So since moving over here, I believe you pretty much stayed on the East Coast. And then, as I said, in 2020, 2021, joined Glue Genes. Glue Solutions, I should say, the company. All right, I'm not going to steal more time. Jonathan, it's a pleasure to have you here. I'm very curious to hear what you have to say.
All right, thank you, Simon. Yeah, so I'm here. I'm going to give a few disclaimers at the beginning, right? So this is a seminar on image analysis and the biologists. I'm very much not a biologist, so I will attempt to give some context of the biology I'm going to talk about, but I'm going to be light on that so that I don't say too many wrong things. And hopefully you guys already know more than I do, could possibly explain to you anyways. And yeah, glue itself it's a general purpose tool for exploratory data analysis. It's not specifically an image analysis tool, although it is definitely motivated by the desire to combine images with other kinds of data, tabular data, higher dimensional data sets as well. And so I hope that you will find that interesting and potentially useful. And whether that's the core kind of glue software, which is this open source Python library, or the specific customizations that we've started to build that we're calling glue genes that sort of are working for genomics analysis. And in particular, today I'm going to talk about its applications to looking at single cell sequencing data and spatial omics. Okay, so start by just showing a picture. This is what the glue genes kind of environment looks like. I'm showing the desktop application here, which is this Qt application you can download. You can load your data sets in, you can link them together, you can make all sorts of figures and combine them in a way that you can selections from one propagate to the other. So just wanted to show the software first, and then we've got a little 2-minute promo kind of set of slides to kind of get you oriented around Glue, the basic software. So we're going to start with that. So what is Glue? It's not an acronym. Astronomers in particular, and I think biologists as well, have a problem with naming everything as acronyms. Glue is not an acronym. It's called Glue because it's open source software that glues together data sets, glues together graphs, and it glues together tools. So what do we mean by data? Well, anything, right? So any sort of data sets that are files in your computer, whether they're tables of numbers or images or higher dimensional data cubes of information. And with those kinds of data, Glue glues together the common attributes in those data sets, right? Figuring out either it figures out or you tell it what the things that are in common between the data files are, and that allows them to make these connections, which allows you to sort of explore and interrogate the relationships between and within your data sets without needing to merge your data files ahead of time. What do we mean when we say that Glue glues together graphs? Well, it's sort of shorthand for a bunch of things, right? Common statistical graphics, scatter plots and histograms, as well as maps and images, 3-dimensional displays, including volumetric rendering, and specialized charts, some of which are included within glue, and some of which you can add because glue is easy to customize and extend. One of the key parts of glue, right, is this exploratory data analysis. And what this means is that when you make selections on one graph in glue, those selections propagate across all the graphs. Right? So you select a bar in your histogram, and it shows you, where those points are distributed across all the views. And that allows for this kind of real-time data exploration and insight. And finally, when we glue together, what do we mean by gluing together tools? Well, glue has this plugin architecture, so you can add in your own customizations and use other people's. It can connect to web services, and it's got a built-in command line, and it's fully scriptable, so you can use it in multiple different ways, which allows for easy customization. So that's glue in a brief nutshell, right? Gluing together data, graphs, and tools. A little bit more, right? You can also save, share, and publish what you learn in glue. We have session files, so when you have a glue session, you can save it and come back to it. You can export your graphics to a bunch of other environments. I'll talk a little bit more at the end of the talk about its integration with the Jupyter environment, but there's other forms of export you can export out of glue. So that's glue, and you can learn more at glueviz.org. And glue itself is supported by grants from NASA, NSF, and the Moore Foundation. So that's that. Oh, and I forgot to say at the beginning, I was going to say, we're a fairly small group here today. If you guys have questions, you should definitely feel free to ask them during my talk. You don't have to feel like you have to wait till the end. And that goes for people online as well. Hopefully Sunil can chime in if somebody has a question online. Okay, so that's kind of the glue library, which you kind of need to understand glue genes. And then glue genes is basically a custom version of the glue library, right? So it's built on top of that library, customized for genomics. So it's got loaders for data files that you might want, some custom viewers, and some specific analysis plugins to facilitate particular kinds of workflows. Because it is this open source Python library, right, and that's designed to be easy to customize, this is sort of a starting point, kind of a base. And we've been developing this software in partnership with the Jackson Laboratory. But one of the things I want you to take away from this talk today is that glue genes is it's still kind of early stages, right? We're building this tool out. We think that there's a lot of commonality between the science that glue was originally sort of built to work on, and the data challenges that are facing sort of modern biology. But we'd love to hear from you guys. We'd love to have more users checking it out, asking for what they want. And if you're inclined to contribute programmatically, perhaps contributing plugins that expand glue and glue genes itself. Okay, so as Simon says in the introduction, I'm originally an astronomer, and glue was originally developed by astronomers for looking at astronomy data. So here we have some astronomy image and some catalog, right? So there's a catalog of stars plotted on top of the image, and then some scatter plot that shows some properties of stars that are totally opaque to anybody who's not a deep astronomer, a histogram. And so the basic sort of promo version of what glue does is allows you to make selections, right? So you select on the image, you're selecting those points in the image as well as the points on the catalog. If you make a selection in the scatter plot, you can see where those points are on the image. The same thing in the histogram. And it's got this built-in terminal, right? So you can, if you have something that you want to do that isn't part of the GUI, you can always interact with the data and all the viewers programmatically through the command line. Okay, so why are we bringing this astronomy software to biology, and why are we making glue genes? And the short answer is because of this kind of commonality in data dimensionality and challenges, right? And so in particular, in biology, there's sort of been this expansion of tools to do robust kind of sequencing of sort of smaller and smaller objects and smaller parts of an image, right? And so on the one hand, you have this kind of what I call it bulk single cell. That's probably not the right term. Single cell sequencing, where you take up some tissue, you throw it into a blender, kind of. You've got all the genes, all the cells tagged, and so you know like what gene expression looks like within each cell, but you don't know where those cells came from, but you can kind of figure out which kind of tissue they came from. And on the other hand, you've got spatially resolved looks at things like gene expression, even protein expression, other kinds of modalities, where you have your section tissue and you can look over that image, and you can say, okay, well at this point, we've got these genes are being expressed, or these proteins are being produced, and so that actually turns out to be remarkably similar to the primary use case that we developed it for in astronomy, which was we had these sort of maps of the sky where we take a spectrum at each point on a grid in the sky, and so the spectrum is like the genome. So it's information about at that point of that position along some third dimension, and wanting to be able to figure out the patterns and the structures of this kind of mapping with a lot of rich information along the third dimension. And the other thing that we saw as an opportunity for bringing glue into this question is that there's no sort of in this field of single cell and spatial, there's no sort of one protocol that does everything you want all the time. At least not that I'm aware of. Maybe you all have your favorite thing that you think does almost everything. But in this particular case, I'm just highlighting 3 of the different protocols from 10x genomics. But basically this sort of stuff applies to nanostring and Merfish and other protocols as well. Right, so you have a choice sometimes, right? So in the case of where you're just doing this sort of bulk single cell analysis, you get lots of signal to noise in your data. You can cover the full genome, but you don't have any spatial resolution. You don't know where in an image, for instance, these data points came from. In a typical kind of spatial transcriptomics kind of environment, you can see the full set of gene expression, for instance. Your spatial resolution, though, is not super high. It's not yet down to the level of like, here's a single cell, here's the gene expression within that cell. So it's a little bit less, of course, than that, a little bit less fine than that. And the data quality is a little bit lower signal to noise, right? Because you don't have, you're limited to sort of that thin slice of tissue, and you don't get quite as much, quite as high quality data. And then you have there's a variety of different sort of in situ kind of analyses where you can get extremely high spatial resolution and good data quality, but you may be limited to tagging specific genes, specific kinds of regions of the genome that you're interested in looking at. And so this sort of puts us in a world where often what you want to do is bring together data sets from different modalities and combine them. Right? And that's kind of what glue is all about, is combining different data sets. Okay, so apologies, I made this slide a while ago before the Titanic sub disaster. So perhaps it's in slightly bad taste, but I'm not going to remake it. So of course we all know about the importance of metadata, right? Metadata is the information about your data that's so important for keeping track of the provenance of your data. And really, you need to spend sort of as much time thinking about the metadata attached to your data as you do about the data itself, right? And so, in an ideal world, we're using these well-defined ontologies, and we've got all these standards that we can follow for our data sets, right? But we're also all kind of busy scientists, right? And our data sets aren't actually that clean and neat with all this well-defined structure around them. They're CSV files scattered across our hard drive with version 2 dash final, no really, right? And that's just life, right? Like sometimes you're going to use some well-defined data sets, sometimes you're going to use some messy data sets. You're going to put them together, And and Glue understands, right? And Glue is here to help, form of a lifeboat, coming to save you from the fact that not all of your data is perfectly defined with your metadata. What do I mean by this? I mean that there are lots of tools that will take nicely defined data sets with all their rich metadata and load them in. But sometimes those connections between data sets, that metadata really only exists in your head, right? So you know that this obscurely named thing in this column is the same as this in another data file, right? But no software can be written to tell the software that is the case, because it's just some name you gave the column, right? So in Glue, if your data comes in with structure and metadata, it will automatically suggest the links between them. And if that's not the case, then you can manually define those relationships, right? So here's, in this case, we've got some spatial omics data that comes in with structure, and then an image that isn't connected structurally, but you can just use this graphical user interface to set up the links that exist. That spatial 0 in this data set means the x coordinate in pixels, right? And so you can set that up. And if it doesn't mean that in pixels, then I'll show you a little bit later. You can easily customize, write like a single line of Python basically to add in whatever custom translation function you need for your particular data files. So for those of you who work in single cell spatial omics type stuff, one analogy I like to give is that Glue is like ANData, only more so. So ANData is this Python package for handling annotated data matrices. It's often used in Python libraries that support this kind of data analysis. And what it does is, you've got this two-dimensional data array that defines, in this case, something like the expression of a particular gene at a particular observation, or in a particular cell, or at a particular point in your image. And then you've got tabular data, tables of data that describe information about those genes, that portion of the genome, and another set of tables that describe information about the point that information was taken at, whether that's a cell or a point on the image. And so that's been a very powerful kind of approach for building tools on top of this. And Glue is kind of like this, only more so. So the same kind of links that exist within ANData are like one kind of link in Glue, one kind of way of linking together data sets. And so there are more. And so we can add on additional stuff that doesn't exactly neatly fit within this paradigm. And you don't have to worry about getting all your data files into an ANData format ahead of time. So I'm going to go through like a little toy analysis here in Glue genes. We're going to be looking at RNA-seq, spatial transcriptomics from some public data set. We're going to imagine, well, not imagine, in this case, we've done some analysis of the data already in some external program. So we've sort of modified it in a way that means that the original software that comes from the device manufacturer doesn't load it back in. And we want to join it to some other data set that we have, right, from a different study. In this case, we're going to be looking at EQTL data for the hippocampus. EQTL is basically, right, it's information about how expression of the genome in one part of the genome is correlated with specific SNPs, specific mutations at some other points. So there are these points in the genome where you make a change and it changes the expression across a bunch of different, across the different genes. So of course you have a couple different options for doing this kind of thing, right? When you want to join some data with some other data set, right? We've got information about these genes that's coming from this QTL data set. These are the same genes we've got the spatial information about. So you could analyze the data sets separately, right? You could look at one and make a list of the interesting kind of genes that you want to look at. You could also, and then, that's sort of an iterative process, right? You make a list in one piece of software, you load it into the other software, and you look at it there. You could also do some merging, right? So if the data files are sort of nice and neat, and you have some experience with programming or merging data sets, right? You could merge the data sets together. And that's a great approach if you know ahead of time sort of which data sets you want to look at, and you only want to do that kind of merging once. But it can be tedious, or it could be hard if the merging process is complicated or whatever, or maybe you just don't know ahead of time which data sets you want to bring in, right? And so the third option, obviously the one we're going to discuss today, is to use glue. So just to be a little bit, a little bit more explicit about this example, right? So here we've got our spatial transcriptomics data set in the central box, and there's this 2D expression matrix, and then these tables of data about the genes and about the observations. Then we've got some image, right? That the image of the tissue that these data were taken from, and so now inside of glue we would say, okay, well, like, these observations correspond to these to pixel coordinates or some other kind of coordinates under some transform, right? So we set up those links, and in this particular example, our QTL data comes in. It's got some gene IDs, ensemble gene IDs, which we can say to glue, oh, these are the same as this column in our spatial data, right? And so these might be different gene IDs. In this case, these are ensemble gene IDs. Here, there's like common names that are the joining key for something else. It doesn't matter, right? Glue does that transitive kind of translation across different kinds of joins. And that's what it would look like in the glue editor. We'd have this sort of web of data sets, and we have a little network visualization which really is mostly useful for helping you see like which data sets don't yet have links to other data sets, because most of the time you're going to want your data within glue to all be kind of linked together. Okay, and so when you set this up, right, then this means that when you make a selection in your QTL data, whether that's using it as a table or in some scatter plot or something, then that selection will propagate to the genes in your spatial data set, propagate to your expression matrix. And from the other direction, it works as well. If you select a portion of your image, you'll select the observations within that portion of your image, which will select the portions of your 2D expression matrix. Okay, and so this is then a little screenshot of that sort of in action, right? So we'll first set up the link that isn't defined in our metadata. And then once we have that link set up, we can make selections. I'm making a selection in the 2D scatter plot. Now we haven't yet added the data to the image, but it's a simple drag and drop to do so. And when you do the region that you've selected there, it's maybe a little hard to see, but there's orange and blue dots on the screen. You can zoom into the image. You can make selections on the image, right? So now we're selecting both the data points and the actual pixels in the image. If we want to look at the actual pixels in the image, we have in the same kind of data set. We've got a bunch of different Boolean tools for doing your subsets, so you can make sort of arbitrary selections. And so here I've done an intersection in the histogram between that spatial selection that we made and the kinds of here we've already sort of classified the kinds of dominant kind of cells present in these particular observations. Maybe I'll just show that just one more time, just because it kind of goes by fast. This is kind of the core glue interaction that sort of is valuable to see, to sort of think about what kind of things could I use this for. And so you can see some of the things, like when you make selections, you can choose to take an existing selection or make a new one. You've got all these different tools for making selections in multiple different ways. They can be pretty arbitrary shapes, and you can use these Boolean tools to kind of do intersections or add additions to subsets.
So, short question: can you also overlay graphics? You have these nice selection tools, but can you define a function and then sort of display that curve or whatever in the image or in the scatter plot? Sort of vector graphics.
So you want to display a...
Maybe you define a function, like, simplest, a circle, right? You just want to plot that circle and see it.
Well, yes, no, maybe not super easily. So you can define your subsets programmatically through the command line. So you can, if you want a subset to be a circle at a particular point with a particular size, then you can define that from the command line for sure. If you have regions of the image that you already know you're interested in, we support a bunch of different sort of region files that you can load in, and then those region files can become subsets. But we don't really support directly like drawing onto the viewers in that way. Although if you know what you're doing, you can do it because they are just matplotlib viewers in the background, right? And they've got their artists, and so you could do it, but it's not like well supported. Yeah.
How efficient is Glue at like handling big chunk of image data, let's say 40 or something like that?
Yeah, great question. It depends, of course, on the data format that underlies the image, right? I'll show you later. We have an example of one of these OME ZARs that we support in Glue, right? So it's like in that example, it's like 36 gigabytes of data, right? And it's multi-resolution, and so it's all it's not all loaded into memory. It loads the piece that you need at the resolution you need, right? So Glue viewers are designed to support very large images because astronomy images are also very large, and so depending on the data format, it's more or less fast and efficient at doing that. Right? There are some data formats in astronomy, anyways, and I'm sure in biology that don't sort of support like partial loading, and sometimes you just have to load that all into memory. But if you do have data files that support that, then Glue will deal with it appropriately. Yeah. Okay. So a brief digression into talking about the value of GUIs and the command lines, right? So people are lazy. I mean, not you guys, but like people are lazy. And plotting syntax between different libraries is kind of complicated and annoying, right? And so the value of having a GUI kind of interface for making plots is that if you make plots easy to make, people will make plots, right? And so Glue's, there's a video. But anyways, the point is that making plots is as simple as dragging and dropping. There's a little pop-up window that asks you what kind of plot you want to make, and there you go. And the other thing is that I think that happens is that using a GUI for configuring plots actually kind of encourages these kind of good accidents that happen when it's a lot of work to figure out which things you want to plot on your image, and you have to figure out the syntax for that ahead of time. Then you probably only make that plot, and you don't accidentally make it with the wrong column. But in this case, you kind of just drag your data set in, and it pops some random, scatter plot of this versus this. And there's any number of times in my kind of usage of Glue where you accidentally click the wrong thing and plot the wrong thing versus the other thing, and you're like, wait a second, those things shouldn't be correlated. Why do they look correlated, right? And so that kind of playfulness with your data is, I think, really encouraged by having a GUI. And so just to demonstrate that, right? Like, I mean, the point is that here that you can configure your plot, right? So here we're showing what we're going to plot. You've got a bunch of different colors options for. In this case, we're doing different categorical variables, and you can put those labels on. You can play with what things are plotted versus size. To the point about big data sets, if your data is really large, the scatter plots, you see like this type density map or scatter plots, like you can plot millions and millions of points as a density scatter plot rather than plotting each one individually. So there are all these different options you can go through, and you can, we hope to provide pretty sensible defaults, and then as well a bunch of options. Okay, so to go back to this example, we're kind of meandering our way through. So if we make a selection of observations or of genes, right, we also probably want to make that link to the other kinds of data, right? And there's not a single correct way to do this. And so glue doesn't like automatically propagate a selection of genes into a selection of cells. What does that even mean? Right? And so you have to tell glue like what that means. And so in this case, right, for instance, if we select a subset of genes, what we probably want to look at in the observations is something like the mean level of expression of that subset of genes in the observations. Maybe it's something else, right? Maybe you have some other metric that you want to define. And so, so this is like the sort of example that we've built out, where, and this is just kind of a schematic picture of that. You know, you make some selection of genes, right? In some data set, either the data set you're looking at, or in this case, the QTL data set, it propagates through. So you get that subset of genes that you're interested in looking at, and then you take the portion of the gene expression matrix that you want to look at, and you calculate some summary statistic, maybe just the mean value of those genes expressed over those cells. And then that summary statistic becomes a piece of information in your observation table that you can use to, say, color code it in a scatter plot or color code it on an image. This is what that looks like in glue. So you've got a little viewer of your QTL type data. You're making some selection of genes there, just to show that you can rename subsets and stuff to keep track of things in glue. And then you have to tell glue how to translate that, right? So there's a couple different functions built in. It adds a new data set. And then when you choose to color code by that, it shows up in the image. And that's live updating. So as you make different subset selections of genes, then that's going to change the color coding in your image, right? And so in this way, you've sort of set up a set of links that says how this subset of information about genes translates to cells, right?
We have a question online. Prazu asks, does glue make it easy to interrogate neural network types data? Example, identifying which image pixels are resulting in strongest activation.
I'm not very familiar with that kind of data. So it is certainly possible that you can, if you have a data set that would show you activation by pixel, then you could look at some subset of pixels and look at that. Yeah, maybe if the questioner could explain a little bit more what that data format looks like, I'd have a better chance of.
Prazu, can you elaborate on your questions, please?
Yeah. Sure. Yeah.
Second question is easy.
Sonali asked, can we load large images, example, whole slide pathology images?
Yes, you can. So it may be the case that not every file format is yet supported. We support loading images through the OpenSlide library. So if your data set can be read into OpenSlide, then you can load it into glue, and you don't load it all into glue, right? You have like this. You can read it into glue, right? So you can read in the correct portions to display the image that you need to, and do the analysis there without having to read the whole thing into memory. And so for purposes of visualization and display, yes. There may be some use cases around if you're trying to analyze the full resolution data, a lot of it, right, that we don't necessarily yet support. I would say that support for like all of the different biology file formats is still a bit of a work in progress, but yeah. Do you have any clarification?
I think Prazu wants to go next.
Okay. Yeah. Sorry, are we?
I think he's typing. I think you should just.
Oh,
Yeah.
all right, we'll just keep going, and we can come back to that question for sure. Okay, and then so that's sort of the one direction between genes and cells, and then you might want to go from cells or observations back again, right? Again, there's a number of different things you might want to do here, right? And there's no one right way to make that connection between a subset over some spatial image and some gene set. So again, we provide sort of one example, which provides a place for somebody to start building their own example. In this case, it's calculating differentially expressed genes between two subsets on the image. And that can be with one subset versus the rest of the image, or two distinct subsets. Right. So again, here in this sort of the single cell case, you select some cells in this dimensionality reduced UMAP view, and then you calculate the genes that are differentially expressed between them, and you set some threshold, and you say, okay, these are the genes that are differentially expressed between these two subsets. Right. And then what that looks like in GLUE, we've got two subsets defined on our brain image. The red and the greens. Apologies if you're colorblind. And we got a little thing that pops up and allows you to specify what the two subsets are and some properties of how you do that calculation. And in this particular case, we do something a little different. We produce a whole different kind of data set that summarizes the information about those differentially expressed genes. It also defines a new subset, which I'm just recoloring here because the brown is ugly. And then you can refine that subset, right? So you can play with things that are more expressed in the hippocampus versus the lateral ventricle. I had to learn the names of the parts of the brain a little bit for doing this example. And so you can sort of see where those points lie now in our QTL diagram. And there's nothing particularly interesting here, just because I was just playing around. I'm not really doing science here, but if there was something interesting here, right, like that's where you would see it. And then of course, just to bring it full circle, right? So then you can go back, right? Like, I mean, this subset of interesting genes can then be plotted on the image, right? So you can color code your points by it. And of course, as you might expect, right now, this is going to have some strong spatial structure. You've literally defined it by which genes are differentially expressed between these two chunks of the brain. So as you would hope, when you select the genes which are differentially expressed in one versus the other, you see the patterns you would expect in the image, either lighting up the hippocampus or lighting up the lateral ventricle. Okay, so a little bit more about some of the features of GLUE genes, right? So we do support a number of different input data types. Actually, the question about OpenSlide isn't on here yet, but we do support that, as well as a bunch of common data files, space ranger output files, ZARs, and then in black, those sort of common generic data files, which we support commonly in GLUE. Yes?
There is two questions online.
Great.
The first one is, does GLUE data normalization method built in, or is it expected that the data is already processed before loading into GLUE software?
Generally speaking, we expect that the data is already processed before we bring it in. Yeah, we don't. We're not a sort of a data reduction setup for specific biology data. I mean, we've afraid there was too many sort of use cases for that, too many ways different people would want to do their things. I will say that I sometimes use it in that way for other data sets, right? Like it is because that process of reducing your data, that might be an astronomy term. I don't know, correcting your data, right? Like normalizing it, figuring out quality assurance, that kind of stuff is really powerful to do in GLUE, because you're often doing some transform of your data, and then you want to check a bunch of diagnostic plots and see what's happening there. So if you're comfortable in Python and working from the command line, then you can do that inside of GLUE. But it's not sort of, that's not kind of not generally built out.
A follow-up question on that one. What can we export out of the software? Plots, images, or visualization?
Yeah, so I don't have a little video showing this, but yeah. So if I go back to a figure, up here in the top left-hand corner of all the viewers, there's a little floppy disk icon, which for those of you who are not as old as I am, is this thing we used to put into computers. Anyways, it stands for saving stuff. So you can save both a Python script to regenerate that figure, as well as the actual image as a PNG or a PDF with a couple different file formats supported. We have Plotly exporters. So if you want to export it as a Plotly figure, that's a JavaScript library that can be easily put on your website. We also support that. We also have another thing that you often want to do out of Glue, right? If you're taking Glue analyses and you need to put them into something else, right? Is you often want to export like the definitions of your subsets or a chunk of your data that's this subset of your data, right? And so there's this export data subsets button up at the top, which allows you to export out a subset definition or a piece of the data around some subset.
Another one, can you name, label different clusters when generating new maps in Glue? How can you set threshold and filtering criteria, or it has to be processed in R?
So if I start this video playing. This is not the main point, so this may be really hard to read. But so this differential genes expression plugin, it's calling ScanPy, which is this Python-based library for doing these kind of analyses, right? So we didn't have to write the code to do differential gene expression with all the thresholds. We just are sort of a wrapper around that underlying library. And so that's kind of the same thing that I'm going to say. Right now, we don't support we don't have a nice GUI plugin for do your own UMAP dimensionality reduction and identify clusters. Like the general expectation is, again, that while you can do that in Glue from the command line, we don't sort of make that super easy. The general idea is you do that data processing outside of Glue, and then Glue is sort of where you bring together that nicely processed data set with some other data that you've also processed, maybe in some other library, right? Like this different kinds of data modalities may come through totally different pipelines, and we don't support all of that. We're sort of the place you bring together your pre-processed data in general, yeah. Okay, so data loaders, viewers, right? You got standard viewers. We've added some that are sort of new for Glue genes, but except for the one that displays QTL data specifically, they're not sort of specifically customized for genomics. It's just where we built them. And then some analysis plugins. I've shown you the differentially expressed genes and the expression of gene subsets. You can also look up KEGG pathways for gene subsets. So these are like almost, right now, they're sort of demonstration plugins, right? Like they were built as we were working with the scientists at Jackson Laboratory because they wanted these particular features, right? But they're just kind of, they're also sort of path like examples for people to, if they want to take and customize and add their own things, right? Whether that's calling some external library, calling some external data set, on the web that they, like the KEGG pathways one, right, uses some library lookup function to go get that information from some third-party service. Okay, so this is the case of large. So I'm going to show just a couple of the other viewers in Glue that I haven't shown so far. This is the case of a large data file view. So we support these multi-resolution views. This is, again, as I said, a 36 gigabyte OME ZAR file. It happens to be living locally on my hard drive here, but it could be the underlying library we use for this is sort of agnostic about whether the file is local or remote. And so here we've got this large file, and we can zoom in. We can scrub through. There's a z-axis here. If there was a time axis, it would also be down there. You can scrub through them. You can just pan around. There's two different kind of channels that we load as different attributes, and that sort of all just kind of works kind of fast and quickly, as glue is only drawing in the information it needs to from the file to display the correct resolution. We have a custom viewer specifically for QTL type data, just because that was requested, and it just kind of shows the different chromosomes, and it's basically a scatter plot with a few extra little niceties. Here's some single cell data, this time from a mouse experiment where there's a bunch of different mice from different strains and different diet conditions. So we've got a little small multiples viewer, right? So now instead of showing, so this is on the left is the full thing, and then on the right is faceted down by these different experimental factors. So you can look for where which of your cells are different between the different strains and the different diets. You can change around those factors. And then we've got heat maps, and there's a little tool to cluster them, right? So you can do hierarchical clustering of your heat map data to look for patterns and make selections in the heat map. Those selections propagate to other data sets. And as I've alluded to several times already in response to questions, Chris Beaumont, who was the original author of Glue and is still with Glue Solutions, sort of wrote this up as kind of this term hackable user interface. The point is that there are some things that are much more natural to do with a GUI. And so the canonical example, right, is to draw an arbitrary selection in some figure or some image, right? It's tedious to script that. And so you should do that in a GUI. And then there are other things that are best done from the command line from programming, right? So some complicated set of Boolean logic, this and this, but not this or this, right? That might be most logically expressed as code. And so you can do that as code if you want to, right? And so the idea is that everything is accessible through these two different ways. That allows everything to be scriptable if you want to script things and set them up. And it allows people who don't know Python, don't know programming, as well to use Glue without missing out. Although there are some of these things that it's like, well, it's not specifically built into Glue, but you can do it. If you're familiar with Python, you can do it by scripting it. And so, just doing a little time check. Yeah, so you can customize Glue at a variety of levels. There's this plugin architecture, right? So there are existing plugins. Glue Genes itself is basically just a set of plugins for the core Glue library. You got a config.py file that's is the sort of easiest and fastest way to hack your own version of Glue. You have this little Python file, and you can define a bunch of custom things there if you need to. And then there's writing your own custom plugins, modifying core Glue, probably not something you normally need to do. So just to say, like, this is we make it try to make it kind of easy for you to customize Glue to work for your own thing, right? So if you want to define a custom linking function between data sets, it can be literally as short as this. Like, you put this in a config.py file that lives in your home directory or where you launch Glue from, and now this linking function will come up in that GUI that allows you to link together different data sets. This is a very trivial example of doing some sort of scaling thing, but they can be more complicated. But sometimes you just need like this little thing, right? Like this little translation. In the same way, custom data loaders, this is a problem that often arises with GUI-based programs, is that it's hard to add new data types because If they're not supported, they're not supported, and then you're stuck, right? But adding a new data loader, again, certainly if there's a Python library that already does the loading, then can be very short to write a little bit, to write your own custom function. If you've got some CSV file that's particularly hard and ornery to get in, and the defaults in Glue don't allow you to load in that CSV file, well, you can just write a little wrapper around this, and it'll pop up as an option to load in that particular CSV file inside of Glue. And then I've shown you all examples from the desktop version of Glue, but sometimes what if your data is living on some remote cluster somewhere, or you want to provide nice interactive web page showing your analysis, and this is where Glue Jupyter comes in. So Glue Core is kind of the base, and there's these Qt type viewers and application, but we also have this whole version called Glue Jupyter, which is running in a Jupyter notebook environment and has sort of JavaScripty viewers. And we're working now on Glue Jupyter Lab, which is sort of a full alternative version of this. It all runs in Jupyter Notebook. Why would we want to use web viewers? Well, there's some very nice web viewers for genomics. This is the Gosling.js library, which has all these beautiful visualizations of genomics that we don't want to have to recreate. But we can borrow these kind of works for in a more sort of web-based environment. This already works in astronomy. We have this is a picture from the James Webb Space Telescope, and the first look data tools for the James Webb Space Telescope run this thing called JDA-Viz, which is based on Glue. And this is one of their sort of sample applications. This runs in a little website on the data archive, so you can take a quick look at the data before choosing what you're going to download. And it also runs in a Jupyter Notebook, so you can start your analysis on the website and then transition to a notebook later. And so this is the thing that currently in progress, currently being built, that is basically, this is now Jupyter Lab, right? And we're kind of using it as a front end for building a web application. So you've got all your viewers, and you can drag them around, and you can customize them within the application. And then you can also pull up a notebook. So if you like working in Jupyter Notebooks, right, you can use a Jupyter Notebook to interact with the Glue session. This is the mode of working in that I really originally wanted for Glue, because if you're working, I like to work in Jupyter Notebooks myself. If I was working in a Jupyter Notebook and I come to a figure and the figure is static, but I really need to be able to make selections and see those selections against other viewers, I don't want to have to save out the stuff from the Jupyter Notebook and load it into Glue. I want just to be able to transform my notebook into a Glue-like session, and that's what we're building here. Okay, so I've talked about kind of these kind of philosophies already. Just to reiterate, we want to connect data sets, we want to make it easy to create figures, we want to make it easy to extend and customize, and we want to make it easy to use in different workflows, so the desktop and the web, like Jupyter views. You can install GlueGenes in two different ways. We have these standalone bundled installers that you can download and just run. And then if you're familiar with Python or you want to play around with it more, right, you can basically do pip install GlueGenes to get your GlueGenes thing in the Python environment and play with it there. Okay, and so that's GlueGenes. Right, it's this customized version of the Glue library, and it's, as I said, it's still under active development, very much so. I'd love to encourage you guys to check it out and give us feedback. Let us know what you think, what you'd want in it, and what bugs you encounter, because there's always bugs in software.
We have some questions online, and we have some in the audience. Let's start with the audience because you showed up.
Hey, really great talk. So I guess as somebody who's worked mostly in the kind of cancer bio space, but has recently been collaborating a little bit with people in the astronomy space, I feel like these two, they're very siloed off, at least in my experience, despite kind of having very similar, I don't know, goals. I'm wondering as somebody who, when you come from the astro space and have now kind of come upon this single cell analysis space, are there I don't know, visualizations, visual encodings, analytical workflows you think that the people in astronomy are doing kind of well that we're not doing here in biology? Are there things that you think we're really missing? Or I guess that's it.
Yeah, that's a great question. I don't know. I mean, some of the challenges are very different, and so some of the things that we do in astronomy are probably not super applicable. The big one that comes to mind is, and one of the things that GLUE is kind of unique about is when we have these three-dimensional data cubes. Sometimes where we really care about paths in them. So like imagine a spiral galaxy. Sorry, that's got a galaxy with spirally arms, and you want information like along those paths. And so extracting like a slice that looks like a two-dimensional slice, but is the flat version of a path along that is something that we do a lot in astronomy. And I think there may be times in biology when you want to do that. And maybe there are already tools to do that, but it's something that comes to mind that I haven't sort of seen as much of.
That could be like a digital biopsy or something like that, where you're like slicing through some.
Right. Yeah. At with some arbitrary shape. It doesn't have to be a flat plane. It can be some complicated shape. Yeah.
All right. Hi. Thank you for your talk. My question relates to, often with biological data, the links between different types of data are probabilistic. So you have one low resolution data set, one high resolution data set, and you may not have a one-to-one correspondence, but there's some kind of probability. In particular, I'm thinking we do like in situ hybridization as well as single cell sequencing. And so there's not a one-to-one correspondence, but ideally you might be able to say if there's a particular cluster in your single cell sequencing that they may show up and, based on your in situ results. How does that work?
That's a hard problem and not one we've sort of exactly tackled yet, right? So when we've been thinking about joining together like a single cell and some spatial stuff, it's mostly been at the level of kind of genes where like those are kind of well-defined-ish things. Although even there can be probabilistic kind of associations if you're talking about like orthology from one species to another. So we don't yet, right? And this is definitely something that comes up in astronomy, and we just haven't quite figured out the best way to handle it. In astronomy, you have like images at different wavelengths, like X-rays and optical, and X-ray is much lower resolution. And so you've got a blob, that's your X-ray blob, and within it you've got multiple optical point sources, and you want to know, which one is which, or should it be split up, right? So at this point, we're still like saying basically that's something you would do in some external analysis tool, and you could bring in your best bets, your best guesses for what that connection should look like into GLUE, and then maybe you could explore it in that, but we don't sort of support all that kind of thing. But we'd like to. I need to figure it out.
Okay, so we're sort of a little bit over the hour. If anybody needs to run, they're excused, but otherwise, I think we can maybe take a few more online questions. There was one, or three in one, I think.
There is 4.
And while we pull those up, I do want to say just in the back, we have some stickers, we got some notepads. If you want to take a little bit of GLUE or GLUE Genes swag home with you to remind you of the talk, please do.
There is a question online that's it. Can we just make the use of GLUE software along with the other viewer, Napari?
Yes, great question. Yeah, I had we had to talk about Napari a while ago here in this session. I prepared a slide in case somebody asked this question. So not yet, but I mean, I think it's interesting. So Napari is a more full-featured image viewer for biology images than what we currently have inside of GLUE. So one way of thinking about that is, could we use just the Napari viewer as one of our image viewers within GLUE? We have an analog in the astronomy world, that's this thing called Worldwide Telescope that allows you to view astronomy images, and we use that as one of the viewers for images inside of GLUE. So maybe someday, not quite yet. GLUE and Napari share a lot of common sort of philosophy and lineage, So it might be fairly easy, but we haven't done it yet.
It's follow-up question, I think, our same question on that one. So can we use GPUs to render 3D volume inside the glue?
Yeah, so the three-dimensional viewers in Glue, both 3D scatter viewer and the 3D volumetric viewer, neither of which I showed here, but we have both of those, and they both use BizPy, which is GPU accelerated on the back end. So yes.
Just one more.
Okay. So we'll take one more. And then, so what we cannot do from a script than a GUI. So
What can't you do from a script that you can do from the GUI?
Yeah.
Nothing. So everything is fully scriptable. There are things you can't do from the GUI that you can do from the script, because from the script it's arbitrary, right? You can write whatever you want. But everything that's in the GUI is scriptable, so you can customize it programmatically if you want.
All right, thank you. I think we stop here. Thank you all for the questions, online and in person. Thank you, Jonathan. It was fantastic.